connect back to the community who has given me so much. So I really appreciate Dr. Mugulkar reaching out to me. There is only just one correction. I work at Penn State College of Medicine. This is not veterinary medicine. Fortunately or unfortunately, I have been stuck with medicine for a while now. So anyway, the topic we are discussing today is gut barrier. So the outline is pretty straightforward. I'm going to introduce a concept about intestinal tight junction barrier. I will discuss a little bit about current projects in my lab as a basis to understand the intestinal tight junction barrier and its role in intestinal inflammation. And then I will go over how several pathogens they interact with tight junctions. And I will make sure we have some time for question and answer session. So there is so much going on this simple, tube or intestine that we always think. So obviously digestion and absorption is the most important function, and you can appreciate here mucosal folds and then villi and crypts. All this goes perfectly. It increases surface area tremendously and goes perfectly with the mission of the gut, that is digestion and absorption. But this also leaves the intestinal surface as the largest surface area that is exposed to environment. This is largest surface area, even larger than the skin. What does it mean? That it is now in touch with environment all the time. And the environment in lumen here is also very harsh. There are food byproducts, food waste, there are toxins and pollutants, and not to mention, millions and trillions of microbes. So there are safeguards so that the intestine can absorb nutrients, but at the same time is not harmed by the harsh luminal environment. So what those safeguards are, there is mucus layer, and there are antimicrobial peptides that are secreted by intestinal epithelial cells. There are immune cells in the lamina propria that keep surveilling the luminal surface. But by far, one of the most common or important barrier that we have against the harmful agents in the lumen is called tight junction barrier. So this is important part of innate immunity that we have in the gut, not in the gut, but even other surfaces, including the respiratory system. So where are these tight junctions located? They are present right in between two cells in the intestinal epithelial surface at the apical side. Obviously, they are not visible in the histology section, but in electron microscope, you can um, appreciate them as where two lateral surfaces of two adjoining cells, they come close to each other. We called it actually a lateral case. That's how the tight junctions will look like. There are actually other junctions also present, something called adherence junctions, obviously helps othering two adjoining cells, and there are gap junctions, which helps communication between two cells. But the tight junction is the primary determinant of the paracellular space. So what can go in through two adjacent cells and what can't pass through. So how the tight junction composition looks like. In here in this diagram, I'm showing few important proteins in the tight junction. It's a complex of large number of proteins. One family is called claudins, and there are several members. Around 25 members of claudins are described in mammals. So these proteins are transmembrane in the sense they will pass through the membrane 4 times, and then they will attach. So one claudin on one cell will attach to the claudin on another cell, and this can be homogeneous association, say claudin 1 on one cell to the claudin 1 on another cell, or it can be heterogeneous. There can be claudin 1 and claudin 3, those kind of things. Another important protein is occludin. Occludin is a very important protein for forming the barrier. And there are other proteins such as JAM. This is immunoglobulin family, junction adhesion molecule. So these transmembrane proteins, they are attached to cytoplasmic plaque proteins called ZO1. ZO1 forms a plaque where all these membrane proteins, they join together. And ultimately this complex is joined to actin cytoskeleton. But this is not so simple because several cloudins, they are barrier forming, but few cloudins, such as cloudin 2, is actually pore forming. So it will form a pore through which sodium and water can pass through. Occludin, on the other hand, is a barrier forming protein. So in most of the inflammatory conditions, cloudin 2 levels are increased. There is increased permeability, and levels of cloudin 2 are decreased, and the barrier is decreased. So these tight junctions have two important functions. One is gauge and another is fence. So gauge refers to the ability of tight junctions to regulate this paracellular permeability. What can pass through this or what can't. And the fence function refers the ability of tight junctions to provide polarity to the intestinal cells. So for example, because of tight junctions, the apical domain is maintained apically and basolateral domain is maintained basolaterally. We know this plasma membrane is fluid, but we want the proteins that are present on basolateral side to be on that side. They shouldn't go to apical side, and proteins present on apical side should not go to basolateral side. So that function is called fence. Now, whenever something goes wrong, the barrier is disrupted, there is increased permeability here that leads to something commonly called as leaky gut. So in leaky gut, it's very common, at least in US, in leaky gut, there is increased paracellular permeability or increased tight junction permeability. So in the lab, actually, I'm focusing on something called inflammatory bowel disease. This is very common condition in US. It was supposed to be a Western disease, but now this is picking up in Asian countries, including India. There are two different forms. One is Crohn's disease and another is ulcerative colitis. Both conditions are very nasty in the sense there is no cure and it affects patients' life very badly. And in spite of a lot of research, we yet don't know how IBD is caused. There are several factors, including microbial antigens, excessive host immune response, environment, genetics, they are involved, but we really don't know, and there is no cure. The current therapies have a lot of secondary effects. So the hypothesis in the lab is that defects in intestinal tight junction barrier leads to intestinal inflammation. So what does it mean? This is how tight junction barrier defects are involved in inflammatory bowel disease. So in homeostasis, there are two pathways. One is pore pathway and another is leak pathway. Pore pathway, as I mentioned on previous slide, is defined by Claudin 2. Leak pathway is defined by occludins. So under normal circumstances, the Treg cells, they secrete anti-inflammatory cytokines, including IL-10 and TGF-beta, which keeps Th1 and Th2 responses under control. But whenever there is inflammation, now there is excessive activation of Th1 that will lead to excessive secretion of TNF alpha, interferon gamma, and that will ultimately activate something called myosin light chain kinase. It will act on actin cytoskeleton and will open up this leak pathway. Similarly, Th2 pathway will secrete a lot of IL-13, which leads to increase in Claudin 2 expression, and that will open a pore pathway. So what is the result? The result is there is excessive now permeability that will lead to luminal antigens to come inside the host, and then this process goes on, leading to unresolved inflammation. Ultimately, intestinal epithelial cells die, and now the whole epithelium is unrestricted. And that's how we think tight junction barrier is important. It prevents luminal antigens to come into the host and cause excessive immune responses. So how do we measure tight junction barrier? There are several ways to do it. In mammalian small intestine, if you induce exogenous current, then resistance is created at few places. In most of the cases, the resistance comes from tight junctions, which are present right at the apical side. If you can measure that resistance, that becomes something called transepithelial resistance, and that gives you fair idea of how intact the tight junction barrier is. And this can be done very easily in cell culture. In cell culture, we grow intestinal epithelial cells, something on some special filters, and then there is this chopstick kind of equipment which is connected to oehometer, and you can simply measure the resistance. So it's very pretty high when the epithelium is intact, the resistance will be higher. When epithelium is damaged, the resistance goes down. Alternatively, you can use something called using chambers. So here in my lab, what we do is these chambers are kind of small chambers where you can actually mount intestinal mucosa, either from mouse or human biopsy or poultry or whatever. And then these chambers are connected to voltage clamp, and you can measure all electrical parameters, including resistance and potential difference and so on. And then this is connected to computer, so you can have real-time data. Another method to do study gut permeability or intestinal permeability is by in vivo recycling perfusion. Here in anesthetized mice, we isolate intestinal loop, cannulate it at two ends, and with a recycling perfusion pump, we perfuse those intestinal loops with probe, for example, fit selectable dextran, for example, and then we calculate how much we started with and how much has gone inside the cell. So it's pretty straightforward. So if the permeability is higher, then we'll lose the probe. So this is how we assess the tight junction barrier function. So in the lab, we are interested in something called autophagy or self-eating. So whenever cells are under stress, cells will degrade its own constituents. It will degrade abnormal protein aggregates or long-lived proteins so that they can recycle the nutrients to cope up with the stress. So in this figure, you can see this is the autophagosome. So all the with some cargo proteins which are now ready for degradation. And this pathway has come into picture in recent years, and the most probably common relation is how autophagy is induced, and one common mechanism is by starvation. We are actually, and you might have heard about intermittent fasting, which is very common nowadays, and we are actually collaborating with Dr. Dixit on this project, because what if any one of you is following that Dixit diet plan, we think intermittent fasting actually induces autophagy, which has several beneficial effects. So in terms of pathway, we'll not go into details, but this is a very elaborate mechanism where several ATG related proteins or ATG proteins or autophagy related proteins, they come together, they form a membrane, and then they engulf the cargo proteins. So these cargo proteins are again, long-lived proteins, misfolded proteins, so that cells can recycle the nutrients. Ultimately, these cargo proteins are engulfed in something called autophagosome. Autophagosome then fuse with lysosome, and ultimately in the autophagal lysosome, the cargo proteins will be degraded. And these are not just cellular contents. This is also important for infectious conditions. Many microbial agents are degraded within the cell using this machinery. For example, salmonella. If the cells are deficient in autophagy, intracellular pathogens, they can survive very well. So what is the relation between autophagy and tight junction barrier? In a most simple way, this is a cell culture data where the most simple way to induce autophagy is by starvation. You just remove amino acids and glucose from the cell culture media, and you induce autophagy. When you induce autophagy, you can see here in the red triangles, the resistance goes up along the time. This is four days of time. And as the resistance goes up, the permeability for urea goes down. So urea doesn't have any transporters on the apical on the cells. So whatever urea is passing through the cells, it has to come through paracellular space. So that is how we measure tight junction barrier in terms of fluxes. So when you induce autophagy, TR goes up, paracellular flux goes down. That means the barrier is enhanced. Two different pathways, pore pathway and leak pathway. Pore pathway is regulated by Claudin 2. So this pathway will not allow movement of small molecules. It will allow movement of small molecules, but it will restrict movement of larger macromolecules. And leak pathway regulated by occludin will allow movement of larger micromolecules. What happens interestingly in this case, as you induce starvation in the cells, the Claudin 2 levels goes down while levels of occludin goes up. So we are reducing the levels of pore forming protein Claudin 2, and we are increasing levels of barrier forming protein occludium. The end result is enhancement of tight junction barrier. And this is very interesting because in our biological system, many times when we do something, we get some result, but then there is another secondary effect. But in this case, there are two different targets, but the end result is same, and that is enhancement of tight junction barrier. So instead of going into nitty-gritty details of data, uh, I will show you this working model. So under homeostatic condition, there are these tight junctions, and we are introducing a new theme in here. Generally, we think that the most regulating mechanism is via gene expression. So tight junction will be regulated by, say, gene changes in gene expression of claudin 2, for example, or occludin. But here, what we are saying that besides genetic expression, these proteins, they keep recirculating via intracellular trafficking. So these proteins will move from membrane, go to the cytoplasm. Here, they can be degraded or they can come back, recycle back to the membrane. And that's how normally cell will regulate this barrier. So this is homeostatic condition. When you induce autophagy, what we are proposing is that the pore forming claudin 2 is now increasingly degraded. It doesn't come back. LC3 is autophagy receptor protein. So we are saying that autophagy will take up claudin 2 and bring it to an endocytic component called clathrin and then further degrade it. And this you can see in this image. In control cell, you can see green color claudin 2, which is present on membrane. As you induce starvation, the claudin moves from the membrane, goes into cytoplasm, and co-localizes with LAM2, which is a lysosomal marker. So you reduce the levels of pore-forming protein, and that way you will reduce the paracellular permeability. And you might remember hearing about this clathrin pits, because clathrin this is the mechanism by which even SARS-2 or COVID-19 viruses, they actually go inside the cells. And for occludin, it's a different story. This is barrier forming protein. And we are saying that once you induce autophagy, we increase the levels of occludin at the barrier. The endocytosis is inhibited. And as a proof of concept, we are taking this into animal models. So this is a DSS colitis is a common model of intestinal inflammation in mouse. And we have prepared a special E-rapper diet. So here we are including rapamycin in the diet. As I mentioned, rapamycin is an inhibitor of mTOR, so it actually induces autophagy. So with this diet, when you induce autophagy, the disease activity is reduced. As you can see here, in histology, this is controlled DSS colitis, and with rapamycin diet, you can see the histology is significantly preserved. So essentially, what we're saying is autophagy is beneficial against intestinal inflammation. That was pharmacological way. In the genetic model, we have mouse where we are actually knocking down ATG7 protein or gene. So these mice, they are deficient in autophagy. Now, if you expose these mice to DSS colitis, then you can see here the intestinal inflammation is exaggerated. There's inflammation not just in mucosal layer, but it goes all the way up to serosal layers. So autophagy, on one hand, induction of autophagy is good for, it prevents intestinal inflammation. On the other hand, if you inhibit autophagy, then it will exaggerate colitis. So in the sake of time, interest of time, I will skip through this slide. We are also interesting, as I mentioned, there are no currently therapies against inflammatory bowel disease. So we are interested in developing novel tools against intestinal inflammation. One such pathway we are targeting is aryl hydrocarbon receptor. This is actually called AHR, is a transcription factor in response to xenobiotics. So whenever there are foreign elements in the gut, they will act as a ligand and they will attach to AHR, which is held in cytoplasm. Once this ligand binds to this receptor, AHR will translocate to nucleus and it will bind to certain sequence in the gene we call this dioxin response element, and it will then have effect on that gene expression. So what we are trying to do here, we actually, through large scale, throughput, we have identified TQ or tocopherol quinone as an AHR agonist. And now we are studying its effect on the barrier, tight junction barrier. As you can see here, these are mRNA levels of several tight junction proteins. As I mentioned, claudin 1, claudin 2, claudin 3, claudin 4, occludin. And what we have seen that TQ actually enhances their expression, mRNA expression, particularly claudin 3 is now our focus. And we are currently working around the promoter region for claudin 3. So this is very exciting because, as I mentioned, there are currently no therapies for IBD. So probably you might be also interested to know that several pathogens, they alter tight junctions as shown in this figure. Say, for example, Vibrio cholerae, it secretes a protein called SOT, or it's a toxic compound. It will disintegrate tight junction proteins. Similarly, common enteric pathogen, rotavirus, the VP8 protein of rotavirus, component of rotavirus, will actually disintegrate tight junction barrier. And then attaching and effacing bacteria, which includes E. coli, they secrete their effector proteins through a system called, we call it type 3 secretion system. And several effector proteins of E. coli, including ESP, F, and G, and MAP, they are known to disintegrate or disrupt tight junction barrier. Similarly, Helicobacter pylori or Salmonella, they are known to have so many effector proteins that will induce tight junction barrier disruption. Salmonella, particularly pathogens of Salmonella, is very elaborately studied. And at the same time, it will activate signaling pathways on basolateral side that will lead to recruitment of immune cells, and then neutrophils under chemotactic response. Neutrophils will actually come out through paracellular space towards the luminal side, and during that process will disrupt everything. The integrity of intestinal mucosa will be destroyed. These are some more specific examples of pathogens interacting with tight junctions, particularly tight junction protein claudins. Hepatitis C virus, for example, which is a common cause of hepatitis, is known to interact with extracellular loop of claudin 1. Here, this is very specific interaction, because if you mutate this extracellular loop of claudin 1, then hepatitis C virus will not be able to come into the cells. Similarly, the enterotoxin for Clostridium perfringens, which is very common important pathogen for veterinary industry, we know this enterotoxin will actually bind to extracellular loop of claudin 3 and claudin 4. This is again very specific reaction. The specific residues have been identified, and this is very interesting process. So those were some examples. In some cases, actually, pathogens, they use tight junction proteins as receptors to get entry into the cell. And one example is reovirus, which causes so many different pathologies. So alpha-1 component of reovirus actually interact with tight junction protein called JAM-A, and that is how it will get into the cell. Similarly, there is another virus called Coxsackie virus. This causes pancreatitis and myocarditis in human beings. This virus will actually also use tight junction protein occludin and then go into the cell. And to this list, we can actually add coronavirus, because at least one coronavirus called porcine epidemic diarrhea virus is known to use the similar strategy to get into the cell. Actually, it uses occludin, a main tight junction protein, as a receptor to get entry into the host cell. So finally, I'm going to give you an example of how we can use this intestinal, physiological knowledge to gain insight into the pathogenesis of enteric pathogens. So this is a study that I did several years back, actually. Here we are studying the mechanisms of diarrhea in case of astroviruses. So astroviruses, they are leading cause of diarrhea in young and immunocompromised patients, for example, children in nursery, or HIV patients or very senior people living in the senior housing. But as well as astroviruses are known to affect so many mammalian and avian hosts. So this particular astrovirus, in this case, we are using Turkey astrovirus, which was isolated from field cases of a syndrome called Polt enteritis mortality syndrome (PEMS) from North Carolina several years back. So this during that syndrome, the turkey severe diarrhea, but that was a complex. There was not just astrovirus; there were other bacteria and other viruses involved in that PEMS syndrome. But anyway, what we knew that time is that turkey astrovirus V2 experimentally induces diarrhea in the absence of inflammation and cell death. So here you're looking at histology. This was controlled turkey jejunum, and this is turkey virus 2 inoculated turkey jejunum. And you can see absolutely there is no difference in histology. Absolutely, there is no inflammation, nothing. So the question was how diarrhea, what is the reason of diarrhea? So we then looked at electron microscopy. Here, this is control, and you're looking at the tip of the one, 2, and 3, and the cells are joined together by tight junctions, and there are microvilli in here. So here, tight junctions are very well defined. But as you can see here in turkey virus infected intestine, there is actin aggregates around these tight junctions, these dense electron dense aggregates. And this happens in the tip of the villi, in the middle of the villi, and as well as in the crypts. So that means astroviruses, they induces disorganization of actin cytoskeleton. Actin cytoskeleton is very thick in this upper belt around the tight junctions. So then we wanted to see if actually turkey virus is involved in that actin rearrangement process. So in this conjugal process, sections, virus is stained in green color, and you can see here in infected group there is a lot of virus present on the villi. F actin is stained in green. In control, you can see nice, fine arrangement of apical F actin. But in case of control infected turkey virus infected intestine, you can see the actin arrangement is kind of different. There are spots of aggregation of actin. And as you can see here, the virus actually localizes to these spots. So virus and actin rearrangement, they actually, you can see here green and yellow producing yellow spots in here at different locations. So that means wherever there was virus, it causes actin reorganization around those spots. So what was the reason of diarrhea? Usually we think in case of diarrhea, there is increased secretion, there is increased chloride secretion into the lumen. And that's how, because that's how we have learned it from common examples, including cholera toxin. So to figure out what's going on in here, we actually use something called that using chambers that I mentioned before. And then we put control and infected intestinal mucosa on those chambers. And then on mucosal side, we put actually radio-labeled sodium or radio-labeled chloride, and then collect samples from the serosal side. So that way you will come to know what is going on and how much sodium is going and how much chloride is moving. All we found was compared to control in turkey astrovirus infected intestine, there is significant less movement of sodium. So that means that astrovirus induces sodium malabsorption. The sodium movement has been reduced. Now, why it would cause diarrhea? Because as more and more sodium is held in lumen, it will hold water and it will increase osmotic pressure in the lumen, and that way it will cause osmotic diarrhea. And this is exactly opposite mechanism to secretory diarrhea, which is caused by cholera toxin. So what is the mechanism of sodium absorption or malabsorption in this case? So usually enterocytes have several mechanisms for sodium absorption. Again, this is a absorptive enterocyte, two different domains, apical domain towards the lumen and basolateral domain towards the blood side. There are at least few important mechanisms on the apical side. One is commonly called as SGLT, sodium glucose co-transport. So this transporter will carry one molecule of glucose and one molecule of sodium at a time into the cell, which is very important mechanism for sodium absorption. Another important of mechanism for sodium absorption is NHE, sodium hydrogen exchanger. So this exchanger will bring in one molecule of sodium inside the cell and in exchange for one proton. And this is a very important mechanism for postprandial sodium absorption. That means after we eat food, uh, the energy are activated, and that's how the sodium is absorbed within the cell. And lastly, the third mechanism is ENaC, electrogenic sodium channels, but these are present mostly in the large intestine, not in the small intestine. And we didn't find any difference in terms of SGLT, so we focused on NHE. And what turned out that this NHE3 is important isoform in mammalian or even turkey intestine. So this exchanger is of course buried in the brush border, but it is connected to the actin cytoskeleton through different several connecting proteins. And that is how this exchanger is held in the brush border. At times and during its regulation, this NHA3 will move into the cytoplasm, and at times it will bring back to the membrane. So that's how it is regulated. So when we pursued this, here in Western blot, you can see the total expression of NHE3 is not changed between control and Turkey-Astrovirus infected intestine. Then we fractionated the membrane so that we can into detergent soluble and insoluble. So detergent soluble fraction represents cytoplasm, while detergent insoluble fraction represents membrane. And you can see here compared to control, in the membrane fraction, detergent insoluble fraction compared to control, there is significantly less amount of less amount of NHE3. So that means NHE3 has moved from membrane to the cytoplasm in the Turkey astrovirus infected intestine, and that's how this is leading to sodium malabsorption. So in summary, we showed that astrovirus 2 induces malabsorptive diarrhea, and that is how it leads to sodium malabsorption. And there was actin rearrangement and redistribution of sodium hydrogen exchanger in astrovirus infected intestinal cells. So this is essentially just an example how we can extrapolate our knowledge of intestinal physiology to find out the pathogens of enteric infections. So we don't have time to discuss all of these things, but essentially the recent research have shown that leaky gut or disruption of tight junction barrier, which I mentioned, is not just associated with intestinal inflammation, for example, inflammatory bowel disease or irritable bowel syndrome or celiac disease, but it is involved in so many different biological processes, including aging, cancer, or neurological conditions, autoimmune diseases, and of course, most importantly, obesity, diabetes, and liver diseases. In most of these cases, now people are trying to relate tight junction barrier or leaky gut or increased intestinal permeability to the disease process. So what is the whole take-home message? Unlike what happens in Vegas, stays in Vegas, what happens in gut never stays in the gut, because whatever happens in the gut has large repercussions in terms of how the whole body works or different systems work. So I will stop there. These are acknowledgements. These are people and help that I have. I was very fortunate to get. Finally, this is my email, and I will try to answer if there are any questions. But if you want to contact me for any reason, for any questions, comments, you are most welcome. Thank you. Thanks for your patient listening.
Thank you, Prashant Nighot, sir. Thank you, Prashant Nighot, sir, for sparing your valuable time. And if there are any few questions we can take, if possible for you.
Yes, of course. Yeah. I save time for question answers.
So, Bhupesh, select some of the questions. Bhupesh, can you listen me?
You're muted, Bhupesh. You're muted again.
Okay.
Yeah.
Thank you very much, sir, for a very elaborative and informative lecture on leaky gut, mucosal immunity, then how pathogen interacts with the tight junction proteins in the intestine. It was very wonderful, sir. There are a few questions, sir, I would like to ask on the behalf of participants. Dr. Mercy, sir, has asked you what is the ideal time for optimum autophagy for fasting time? What would be the ideal fasting time so that there should be a maximum autophagy?
Okay, that's a very, very interesting question. There is no definite answer right now. We are actually, say, for example, the model that Dr. Dikshit has shown, intermittent fasting. So you don't eat for several years in a row. That seems to work, but we haven't yet confirmed yet if that induces autophagy or not. That of course has very beneficial effects on the overall health. So in mice, because that's how we come to this knowledge. We go to mice. So right now, what we are doing in mice is we are using a regimen of 16 hours of fasting and 8 hours of food. Um, and it looks like it is working. It is giving us, so we can see actually it induces autophagy in the intestinal epithelial cells, and that's what our interest is. Of course, it will be long before long, time that we realize and we study all the health benefits. But at least in terms of intestinal barrier, what we have seen that, and these are very recent studies that we did in last few months. So when we use 7 days of period, every day, 16 hours of fasting, 8 hours of food, and after 7 days, we put them, on using chambers and do several other things, and we found that it number one, it induces autophagy, and number two, it actually increases or enhances tight junction barrier and reduces intestinal permeability. So that's what the range that we are looking at right now. I hope that's the best answer. I mean, at least I can give right
Yes,
now.
yes, yes. Thank you, sir. Another question is that do autophagy has effect on extracellular organisms and parasites?
Yes, yes, because so far last actually a decade, autophagy work has been focused on immune cells. So most of the knowledge about autophagy right now we have that comes from um, immune cells. So, for example, if you induce autophagy, macrophages they will secrete anti-inflammatory cytokines. But
Okay,
if you make macrophages deficient of autophagy, then they will secrete excessive amount of um, IL-1 beta, for example. So that leads to excessive inflammation. But at the same time, there are several other extracellular factors that are touched by or that are affected by autophagy, including, say, exosomes, for example, and several other these cytokines. So definitely, autophagy has impact on extracellular pathogens or whatever you are looking at.
parasites. Okay, thank you, sir. One of the participant has asked, sir, what is basic autophagy, microautophagy, and chaperone-mediated autophagy?
So what I talked about is macroautophagy. Uh, the chaperone mediated autophagy is a very specific phenomenon. We are not studying it right now because most of the time it is the macro autophagy that is being used or being studied. On top of that, now traditionally people use macroautophagy as a very umbrella term. but in many cases it is more specific, and now people are showing that it's a selective autophagy, in the sense like the selective autophagy in mitochondria, and they call it mitophagy. So we are, previously it was thought that it's a degradation pathway in terms of bulk amount. So when the cells are under stress, they will degrade their components in bulk, and that's how they will recycle their energy. But now, say for example, if you remember the data that I showed, it will specifically degrade claudin 2. So it is a more selective phenomena now.
Okay, thank you, sir. How is methods of detection of autophagy, and is there any biomarkers to detect autophagy?
Oh, such a nice question. So right now in experimental system, we use something called LC3, which is ATG8. It gets converted from LC3 1 to 2. So that conversion is considered as a autophagy inducing marker. And besides that, there are several other receptors that are degraded during autophagic process. So you look at those protein expression of, say, for example, P65. Those are involved in formation of autophagosome and their degradation. So their levels can be monitored as a biomarker. But we do not know, we do not have right now a biomarker that, say, for example,